In this session, we will be trying to predict secondary structure from primary structure. That is, we want to take a protein sequence and predict its secondary structure, meaning predict alpha helices and beta strands from just the sequence itself. It's a difficult problem. Our objectives include being able to distinguish between alpha helices and beta strands and explain what they are, and then take a sequence and predict its secondary structure. Predict where the alpha helices and the beta strands in that sequence would be located. We will specifically look at two algorithms, HNN and PhD, and then learn how to analyze the results. Starting with alpha helices, they are the most abundant form of secondary structure. You can see that it sort of resembles telephone wire or sort of a twisted ribbon. If you notice in the picture, you see that the amino acids follow a sort of twisted coil pattern, and there is hydrogen bonding between amino acids within that pattern. There are about 3.5 amino acids per turn of the helix. The average length of an alpha helix is about 10 amino acids, but they can be as short as 5 or as long as 40 amino acids. Typically, the amino acids that face into the interior of the coil tend to be hydrophobic because they are not exposed to solvent. And one pattern is that the third of every 4 amino acids is hydrophobic. It's a very common secondary structure. And the other common type is the beta sheet or beta strand. The sheet is a series of strands. So the strands kind of make up the beta sheets. So we will actually be predicting beta strands. And it's kind of a pleated structure to the sheet, and there are two types. There is the parallel, meaning that the N to C terminal direction is the same, and the more common anti-parallel, where the chains run opposite. And basically all that means is that there's a little bit different chemistry when they run parallel or anti-parallel, but it's very similar in concept and structure. Okay, so here are the sequences that we'll be using to basically test the algorithms. We're cheating a little bit. The typical goal of a secondary structure prediction is to take a completely unknown protein and predict where the secondary structure is, where the alpha helices are, where the beta strands are. However, we're going to test the algorithms. We're going to use two different proteins. The plant protein is known to have 8 beta strands. And the second protein is a protein that contains what is called the histone fold, a very common fold in nature, and it's known to have 6 alpha helices. The two algorithms we will look at are HNN. HNN stands for Hierarchical Neural Network. And PhD. PhD actually comes from Profile Network from Heidelberg, the HD for Heidelberg. Okay, now here's a web server that runs the HNN program. This can be run locally or through some other prediction programs. And we will start with the plant protein. It's known as plant protein 8 beta strands. And I'm going to paste it into the window, but I'm actually going to use the raw format because I know that this particular program doesn't like the descriptive line. So I'm going to put just the amino acids in, everything but that top descriptive line. And submit. And here are the results. And we see that at the top, the amino acid sequence, we see hashes for every 10 amino acids. And we see a bunch of C's, E's, and H's below. And if you looked at that without knowing what those C's, E's, and H's meant, you'd probably be really lost. Well, there's a bit of a cheat sheet here. The alpha helix is H, and this notation is commonly used for alpha helix and beta strand. H is for helix, and for some reason, E is used for beta strand. It comes from extended strand, and I'm not exactly sure why that stuck, but it did. So almost all of these algorithms will predict E for beta strand and H for alpha helix. Some also predict loops and coils, but we can think of loops and coils as anything that is not alpha helix or beta strand. So typically what we are looking for are the alpha helix and beta strand locations. What we see here is a stretch of E's. That represents 1 beta strand, and another stretch of E's represent 2. So if I count those stretches, I will see 1, 2, 3, 4, a short one, 5, and 6. Now we know because we happen to know the structure of this particular protein. Again, we are kind of testing this algorithm. We know exactly where the beta strand locations are. So let's see, how well did they do? The actual first beta strand goes from amino acid 3 to amino acid 15. And that would be right around here. So it sort of missed the location. It kind of got one, but not quite in the right location. It sort of predicted one in the vicinity. The second goes from 29 to 32, and that would be here. And it actually predicted alpha helix there, so it missed. Third beta strand goes from 37 to 39. Well, it got that one right. The next one goes from 51 to 61, and it really missed that one. So what you really see is that these algorithms are not 100% accurate. So there is still a lot of work to be done in the secondary structure prediction area. We know that the amino acid sequence determines the secondary structure, but we don't know how to predict very well based on it. We do see that the prediction is 24% beta strand and 12% alpha helix. So it predicted that it's more beta strand than alpha helix, so it got that right. And some of the locations are right, but some of them are not. But it did predict with some degree of accuracy that there are beta strands in this protein. Now let's look at the PhD algorithm. This time we'll take the histone fold protein, which we know has 6 alpha helices, so let's submit that one. Again, we're not going to take the top descriptive line, just the amino acids. Okay, well, the good news is you see a lot of blue H's. That means that it predicted successfully that this is a mostly alpha helical protein. You notice there are some uppercase and lowercase letters. For instance, you see the uppercase, then the lowercase sort of flanks that region. What it means is that there is an alpha helix in this region. It could be as short as the stretch of uppercase or as long as the stretch of both. So it's kind of difficult to determine exactly where it begins and ends. So that's the idea behind the uppercase and lowercase. However, for the second one, it's pretty confident in the prediction. It's uppercase all the way, so that's a strong prediction, knowing where it starts and ends. There is a third helix here that wraps around from about 65 to, about 91, then a fourth, and then a fifth, and then a sixth. So it predicted correctly that there are 6 alpha helices. Again, we know the structure of this particular protein, so we know exactly where the alpha helices are. The first alpha helix goes from position 5 to 16, so that goes from about here to here. They pretty much got that right. The second one goes from 25 to 49, and it really nailed that one. Third goes from 64 to 74. Looks like the prediction was a bit longer. Then 82 to 90. Well, that's about here, so really there should be 2 helices here, but it predicted one long one. And then 101 to 125. 101 is where the 30 is, so 70 plus 30. So really that's one long helix in actuality instead of 2 shorter ones. Finally, 134 to 147 did pretty well there. So this prediction is a little bit better, and I want to emphasize, this is not that PhD is necessarily better than HNN. It happens that this protein was a lot easier to predict. If you flip-flop the proteins and run this helical protein at HNN, it would have done pretty well. And PhD would have done equally badly with that beta strand protein. So feel free to give some of these algorithms a try. Look for protein sequences to analyze and predict secondary structure. So good luck with your predictions.